The final speaker of our session is Professor Mohamad Habes from UT Health in San Antonio. Mohamad, over to you.
All right. Thank you, James, and thank you, everyone, for staying to the end. My name is Mohamad Habes. I'm Assistant Professor of Radiology at the University of Texas Health San Antonio, where I'm leading the Neuroimage Analytics Laboratory. I'm also leading the imaging core of our South Texas Alzheimer's Disease Research Center. Today, I'm going also to talk about explainable AI methods and our findings in detecting Alzheimer's disease patterns and how did we do to validate them. So the objective today is to explore the application of explainable AI techniques in early detection of Alzheimer's disease and to put emphasis on the validation so that our models are robust. And how can we do that validation through meta-analysis and ongoing work in our lab using post-mortem MRI data. So we have here today the term black box a lot, and a lot of people who are less enthusiastic about the use of deep learning criticize deep learning networks that they are a black box. And in part, that's true. We have millions of nodes, we have millions of weights, and it might be difficult to look into that network. But once we are able to understand the decision of the neural network, we can build a trust. And once we trust the model, we can take that model to explore new data sets and maybe make new discoveries. And I hope that I can deliver these 2 points to you today through the talk. All right, so how can we build the trust? How can we validate the output of, or the decision of the deep learning network? One way is to use techniques for visual interpretation, and a heat maps is a widely used technique in the field of computer vision. A heat map reflects how much each pixel in a picture or voxel in an MR scan can contribute to the overall prediction of the deep learning model. So assume we have a model that is trained to predict natural writing images like what you see here, number 7. And the neural network can detect among various kind of numbers, this is the number 7. The heat map goes the other way around. From a label, we go back and generate the map, and we know now which pixels have contributed most to the decision. Now, with natural imaging data, we have a problem. Yes, we have a lot of data when we are using platforms like Facebook or Amazon, but we don't have really a ground truth. There are various ways to quantify heat maps. We have here multiple methods today, but we don't have really a reliable ground truth. And Sophie has spoken today about one way to establish a ground truth. We have also another way through the use of meta-analysis. In the neuroimaging field, we have, for example, for Alzheimer's disease, a body of literature that has already looked into what atrophy patterns are present in people with Alzheimer's disease. So when we go and look into the literature, we can summarize that knowledge in a meta-analysis map, and I will be describing that a little bit. So the meta-analysis platform that we have used is the Brain Map portal. The Brain Map portal is being developed in our institution and being led by Dr. Peter Fox for 2 decades in collaboration with Simon Eickhoff from ULISH. And the Brain Map basically has 2 major components. The first component is the Sleuth app, in which you can look for various papers in a given field. In our case here is Alzheimer's disease. So what papers have been published so far to look into voxel-based morphometry and T1-related patterns of Alzheimer's disease, so Sleuth can find those papers for us. They need a kind of decoding in a certain coordinating system, but that's what the Brain Map team is doing. And then another tool called Ginger Ale is summarizing all these maps that have been published across hundreds of studies in a single map that can be used then for validation. And I will be describing how did we do that soon. So this is Dee Wang, the PhD student in my lab who has led all this work and who has published actually his way of validating the interpretive heat maps that we have derived. What he has done is first to conduct a comprehensive meta-analysis. He found, looking into the literature, more than 77 papers 2 years ago. I think there might be more papers now, which have used T1 imaging data and voxel-based morphometry to characterize atrophy patterns with Alzheimer's disease. He has then summarized all these publications in a single ALE map. And then we have built, using the ADNI cohort, a classification model, models, using CNN basically, and classified Alzheimer's disease patients versus controls. We have reached great accuracy in terms of classification and compared deep learning to traditional methods like support vector machines. Then we have used various methods to generate heat maps like LRP, layer-wise relevance propagation, integrated gradients, as well as GGC, guided grad-CAM. And as you see here, these methods generate a little bit different maps between each other. Some of them are more sparse, others are more smooth. And in the end, we have compared our ground truth, which is resulting from the meta-analysis, with these heat maps, the 3 heat maps, and using various matrices. The most important metric maybe is the DICE coefficient, which is quantifying the amount of overlap between the heat map, the ground truth, and that's what Dee has found when we have compared with the meta-analysis map of 77 studies. The bottom row is the map of the meta-analysis, quite expected patterns of atrophy in the hippocampal regions. The first 3 maps are the deep learning maps, and the 4th map is the support vector machine coefficient maps. One interesting finding that we have is that the deep neural networks have higher accuracy in predicting Alzheimer's disease compared to support vector machines, but also they have higher DICE overlap with the meta-analysis, as we can see here in these curves. We have generated these curves through thresholding the maps by dividing by 50, I guess, the max value of every heat map, which has allowed us to generate those curves. Now, the first finding, deep learning networks have higher overlap than support vector machines. And the second finding, among the different heat maps methods, IG was the best, although it was head-to-head in comparison to GGC. So this is the paper that appeared last year online in a neuroimage, and it has a lot of exciting experiments. We had shown that you could synthesize the data, for example, and if the effect in the imaging data is already known, we could reach an overlap, DICE overlap of up to 85%, while in the ADNI cohort, we reach an overlap just up to 60%. All right, now we have validated our model. IG is the best way to go, is the most robust heat map, it seems. Can we take these heat maps to make discovery? What about dementia subtypes? And I'm happy that Mark has started talking about the subtypes. Often, Alzheimer's disease is not present alone in the brain. So this is Carl Lee, a postdoctoral fellow in my lab, has answered the question how clinical diagnosis in vivo would correspond to pathological diagnosis post-mortem. So we went to our brain bank in our ADRC, which had at the time 71 brains. We looked into the clinical diagnosis to the right and compared it with the post-mortem diagnosis to the left. And often clinical diagnosis is biased towards a single diagnosis. Alzheimer's disease, for example, is rarely present alone in the brain. Other neurodegenerative disorders are there. Cerebrovascular disease, dementia with Lewy body, frontotemporal dementias, and so on. So we took a cohort from the NAC database with post-mortem evaluation, and we have built again deep neural networks to classify patients with Alzheimer's disease versus vascular dementia and dementia with Lewy bodies. We focused on these three dementias just because the number of cases is much lower for other dementias. And we have used the IG method to get our heat maps, and excitingly, the deep learning heat maps can show distinctive patterns between the various dementias here. Alzheimer's disease patterns are more focused on the hippocampal regions, while vascular dementia is having atrophy in the hippocampal area, but also white matter disease. Dementia with Lewy body has a kind of posterior occipital pattern. The support vector machines, on the other hand, did not distinguish really between these dementias in terms of pattern, and their classification accuracy was much worse. So this is a paper is now in submission by D, and hopefully we will read it soon. Now, there are not so many publications on these different dementia subtypes, so their patterns, as mentioned, may be the first to be reported now by our group. So how can we validate that? How can we make a validation? Maybe through histopathology and through the use of our unique repository in South Texas, Alzheimer's Disease Research Center. This is a paper from Carl Lee, who is, again, at the postdoc in our lab, who has published the protocol of the postmortem MR imaging for 300 subjects that are now present in our database. Basically, our brain bank is accepting brain donations. And this is just briefly the workflow of taking the brains, fixing them within 4 weeks, and then do an IA guideline-based neuropathology, and then while the brain is being imaged with post-mortem MRI protocol for 12 hours, we are able to identify abnormalities on those post-mortem MR scans and then guide the sample collection for additional histopathology evaluation. And this is Mariam Mojtabai, a PhD student in my lab, who is working on MR-guided histopathology. She is particularly interested in the evaluation of the intensity of T2 scans and white matter hyperintensities, that those are hallmarks of vascular dementia. And the way that she is doing, after collecting the samples, we are taking histopathological stains. In this example here, we are focusing just on LFP and H and E, although there are much more stains being conducted. And as a result, we are able to tell and validate the MR signal with the histopathological substrates that can be present in that particular location. The same way, we are going to start exploring the interpretive heat maps and guide histopathology, the heat maps that we have just discovered earlier, and guide histopathology with various stains for validation. All right, to summarize, I hope that we were able to convince you that explainable AI methods are getting more attention, and that validation of the heat maps is critical to build the trust in the models and take them to make discovery. Explainable AI methods can provide new insights on biological mechanisms, and finally, we are working towards the use of post-mortem MR for additional validation. Meta-analysis is another way for validation. And with that, I would like to thank our cowboys and cowgirls from Texas. This is our lab team members and our funding agencies. And I would like to thank you, of course.
Thank you, Mohamad. Brilliant talk. So we've got quite a few questions here. I'll start with this. This is a nice one, which I assume wasn't posted by Mark, but if it was, it's very subtle. How do you address mixed pathology when evaluating explanations with post-mortem analysis? Do you think Mark's idea could help to disentangle which features correspond to different dementias?
Yes, I wished I knew about Mark's approach before before this talk, but so far we have been using classification models that are considering not just one single label, but multiple labels. So this is exactly how we have described that in our paper.
Right, great. So a specific question about the ginger ale analysis you mentioned. Do you account for at all for differences in pre-processing and kind of participant inclusion criteria when you're pooling the data across those different studies?
Yes, that's a great question. I guess there are guidelines for the use of ale and ginger ale, and I guess if the study does not have sufficient power, we don't include it. So that's one of the criteria for filtering the studies to be included in the final meta-analysis or not.
Okay, and then another question. Are there any, do you know any meta-analysis resources like BrainMap and Neurosynth for functional connectivity? And could you provide a ground truth on connection level instead of for regions?
Well, yes, I mean, the BrainMap can do that. The BrainMap, I have shown here examples of VBM-based meta-analysis and T1-based patterns for Alzheimer's prediction. But the BrainMap can also generate maps for functional MRI. We have worked for amyloid, for example, and I appreciate Sophie's approach for generating a ground truth. There are not so many papers published on amyloid patterns or functional MRI patterns in Alzheimer's disease, so it's really difficult to create a meta-analysis when there are no data, no publications there. So while for T1 we have more sufficient data to build a meta-analysis and create a ground truth, there are still areas where there are either the modality does not have enough publications, or the disease is rare enough, like frontotemporal dementia, where other methods for validation might be needed, like post-mortem evaluation.
Okay, so I'm just going to finish with a couple of questions that actually I'll open up to everyone, all the speakers. So, are black box models inevitable? Why can't we use easier models that are explainable from the beginning? Don't know if any of you guys, I'm asking that question so I don't have to answer it, basically. Why don't we do something easier? Grab the mic.
Is it on? Okay. So, I mean, I think that question is directly tied to the complexity of the model, right? So it really depends on how complex we want the model to be. And given that we can't, or like it's really hard to force a model to encode information in a way that is trivially interpretable by what human designed concepts, like a complex model is to some extent going to be intrinsically hard to interpret. So I don't know to what extent you can really circumvent that, apart from the fact that I think for the most questions you probably just don't need very complex or deep models, and I think it's it's quite controversial for which questions and to what extent you really need deep neural networks for simple questions. And I think it's very reasonable that for many of these questions, simple linear models will suffice, and probably then the better idea is to use them and to keep using them.
Yep. Okay, then final question then. Again, this is for all of you. What effect does redundancy in the brain have on explanations? For example, hippocampus alone might be sufficient to make the predictions. So does the model ignore, or do any of these models ignore other brain regions if you have a really strong predictor? Again, I'm leaving the difficult questions for other people, so.
So in our case, we really use the whole brain signal, I mean the whole MR scan, and all, even without skull stripping, I would say. The model in the end really focuses on the regions that have contributed most to the classification, and as we have seen in the maps between Alzheimer's disease versus vascular dementia, Alzheimer's was more hippocampal focal, while vascular dementia had more widespread white matter disease.
I was just going to add on to that I think this is where some methods can be more useful than others, especially as there are some methods which can show you the direction of importance as well, and show you whether it's contributing more positively or more negatively, and give you a bit more quantification on that. So perhaps if you want to understand whether it's just hippocampus or if there are other regions that are contributing, you could consider an explanation technique that gives you a bit more information.
Finally, I think this is mainly a question about causality, right? And I think it's really important to distinguish between the model explanation and the causal effect that lead from the brain to the actual disease.
Yeah, quite agree. Well, so I think from my perspective, this has been a really great session. We've had MEG, we've had PET, we've had in vivo MRI, ex vivo MRI, we've had histopathology, we've had lots of different explanation methods, and hopefully you guys out there have enjoyed it. So thanks very much for coming, and if we can thank our speakers again.